The journey from genes to proteins, transcription to translation. Have you ever wondered how our body knows which proteins to synthesize in our cells? The answer lies in our DNA, the molecule that carries the genetic blueprint throughout life. The sequence of four bases, adenine, guanine, cytosine, and thymine, forms a unique code that determines the order of amino acids in every protein. Each gene provides a specific instruction to build one or more proteins. Protein synthesis happens in two major steps: transcription and translation. In transcription, DNA is copied into an mRNA. In translation, the mRNA is read by the ribosomes. Amino acids form polypeptides, and those polypeptides make up the functional proteins. Transcription occurs inside the nucleus, where the RNA polymerase binds to the DNA and copies the strand into a complementary mRNA molecule. This happens in three stages: initiation, elongation, and termination. Once formed, the mRNA leaves the nucleus and moves to the ribosome for translation.
The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands.
So this video
The
shows
yellow
process
chain
of
snaking out of the
transcription.
top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA.
Now let's talk about translation. So translation takes place in the cytoplasm at the ribosome. And the ribosome reads out the mRNA in groups of three bases called codons. tRNA, or transfer RNA, brings the correct amino acids to match each codon. The amino acids are joined by peptide bonds to form polypeptides. When the stop codon is reached, the completed chain is released and formed into a functional protein. Now here's the video on translation.
When the RNA copy is complete, all the components of a molecular machine lock together around the RNA, called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules bring each amino acid to the ribosome. There are different transfer molecules for each of the 20 amino acids. The code for each amino acid is read off. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. And after a few seconds, the assembled protein starts to emerge from the ribosome.
So the genetic code is universal for everyone, and it defines how each codon corresponds to a specific amino acid. For example, like the amino acid glycine, it can be coded for guanine, 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 adenine, guanine, guanine, cytosine, and guanine, guanine, uracil. So the proteins we eat are broken down into amino acids, which are reused by the body to make new proteins. Some amino acids are essential or indispensable and must come from our diet. Other non-essential amino acids or dispensable amino acids can be made from the body. Amino acids are the basic building blocks for protein synthesis. Nutrients play a key role in regulating protein synthesis. Vitamin A supports immune cell production, vitamin D regulates calcium transfer proteins, polyunsaturated fatty acids affects genes involved in lipid metabolism, like the hormone-sensitive lipase. This is how genes are controlled by our nutrition. So let's talk about common misconceptions about proteins. First of all, proteins are harmful to your kidneys. Well, there's no evidence that dietary proteins harms the kidneys of otherwise healthy individuals. The other one is that you do not need protein if you're not physically active. Well, you do need proteins for various physiological processes like cell signaling, satiety, and thermogenesis and glycemic regulation. To conclude, protein synthesis is a fundamental process through which genetic information in DNA is used to build proteins. These proteins are important for metabolism, repair, growth, and other body processes. Genes, nutrients, and cellular processes all work together to make protein synthesis possible, thanks to the NFS 284 teaching team and the textbook by Dr. Gerfunkel.